All right. So last time we talked about mapping. So before the exam, we were talking about setting up genetic crosses between flies and mapping, and we were talking about genetic mapping. That is, how often is there a recombination event that happens between two genes, right? I want to segue and then start talking about human mapping. How do we do human genetics and locate human traits? So we're going to talk about the difference between genetic mapping, what we've been doing before, and physical mapping. And we're going to talk about doing this in the context of humans. So the goal in human genetics is going to be to try and identify where in the genome a certain trait is determined, right? Where the gene is that a certain trait or certain disease, it's typically the traits we're looking for as malformities or deficiencies or something like that. The problem in human genetics is you can't do the conventional methods you did in flies. If you're going to do genetic mapping in flies, well, I breed a heterozygote and I cross her to a recessive for all traits male, right? Well, in human genetics, you can't do that, right? You can't use conventional genetic methods. You can't force people to mate with a homozygous recessive male that you've got kept, right? There's lots of constraints about human genetics. So number one, you can't force matings. And humans have relatively few offspring, right? As you've been culturing your flies, you may start seeing you're just getting, hundreds of flies starting to pop up in your vials, right? Because flies are just laying eggs. The females are just constantly laying eggs, and she can have, hundreds, thousands of offspring in her life, right? It's a pretty short lifespan that a fly has. Now in humans, the incubation time is long. You typically only have one child after that long incubation. Like twinning is rare. Triplets and quadruplets didn't really start showing up until we started doing in vitro fertilization and therapies for infertility. So single offspring, 9 months apart, usually somewhere between a year and 2 years between offspring, and then humans just don't have that many offspring to begin with, right? So even big families, you're still only getting into dozens of offspring, and you get a dozen of offspring from a female in 1 week in a fly, right? So this makes it difficult. You have to use different techniques. So
is the Jesse's period for apes similar?
Yeah, I'm not sure. I think it's a little bit shorter in chimps. I don't know. I think it might even be longer in some species, like the great apes might actually be longer. I'm not sure.
I was wondering because I was thinking about the mutations that arise and how you have to have generations after generations to really allow for change to take place. And I was just thinking, for 9 months, we don't have a lot of offspring. There's not a lot of variation.
Yeah, not a lot of offspring in the individual's time span. Yeah, so. Yeah, which is one of the things too. It means the human genome doesn't vary much between individuals, right? If you're doing Drosophila or bacteria or viral genetics, you've got thousands of alleles because they're reproducing so quickly, and there's all these alleles floating around the population. In human populations, you don't have very many differences between individuals, and that's probably partially because of the complexity of us, and we don't tolerate mutations very well, but then also we just have very few offspring, so there's not a lot of chance for mutations and variations to arise. So yeah, you're right, that's another limitation. So you can't look at large offsprings, and like I said, you can't do test cross, right? You can't force matings, nor can you force people with certain traits to mate, right? Now what you can do, and the techniques that you're gonna have to manipulate, is number one, you can take good genealogical records. So we're going to rely heavily upon what has happened in the past, right? You can't set up the cross that you want to do, but you can go back in history and keep good genealogical records and watch how a trait has been inherited over generations, okay? We can take DNA and we can sample people. And we now have the Human Genome Project. So we have tools available, and you can do human genetics. It's just more difficult. So number one, let's talk about pedigree analysis, right? You're going to have to keep good genealogical records. And there are certain populations that are more amenable to doing human genetics. It's usually small populations, which seems counterintuitive, right? Because we're trying to look at a bunch of alleles, and so you'd think a bigger population would be better. Actually, if you've got fewer alleles to consider, that's better, right? Fewer alleles, the better your mapping is going to be. If you've got dozens of alleles, it's going to be hard to keep track of them all. That's what's great about Drosophila, right? You can get thousands of offspring, lots of offspring, from just a single mating event. So there's only 4 possible alleles in a fly cross. You've got a male that might have 2 alleles of one gene, female might have 2 alleles of the other. So you've got a total of 4. You can get thousands of offspring out of that single mating. So you want to keep alleles low and population levels high. So you want relatively small population of people, but they have a lot of offspring. So there's very few populations in the world that do that. One of them that's really good, island populations are really helpful. So if you have a secluded population of people, Iceland, there's a lot of genetics that go on in diseases that affect populations in Iceland. Because it's on an island, there's not a lot of new alleles coming in, it's a relatively small population. The Amish are actually a really good population to study genetics. They're just living in Pennsylvania or in the northern part of the US primarily. And they're not geographically isolated, they're not on an island, that's not what's constraining who marries who, but the Amish have a very tight community, right? And they usually only are going to mate with, marry people that are within their community. So it ends up being a functionally very small population. And the Amish tend to have a lot of children as well. So Amish populations are really nice. Yeah?
So basically the ideal population would be one that follows RD1, right?
Mm-hmm. Yeah, the question was, an ideal population would be one that conforms most closely to Hardy-Weinberg. Yeah, small number of alleles, no breeding from outside the population, no migration events. Yeah. So if you remember Hardy-Weinberg. So Iceland, Amish, and actually the Mormons are really helpful in human genetics. Because for religious reasons, the Mormons want to baptize, they do surrogate baptisms, right? They will baptize a member of the Mormon congregation on behalf of somebody else, because their theology says if you've been surrogate baptized, then you have an opportunity to accept the gospel in the afterlife. So they baptize surrogates on behalf of generations of people who have been dead for a long time. So it's very important for them to try and figure out who's related to who's been baptized, and so they actually keep very good genealogical records. And so there are certain instances where the Mormons release their genealogical records, and they're keeping genealogies of people who are not related to them at all, right? And so they might have a really good genealogy of a population that has a certain trait that you're interested in mapping. So you can contact the Mormons, say, I'd like to see your genealogical records, so that you can do a pedigree analysis and try and analyze how that trait's being inherited, right? So Iceland, Amish, and Mormon are kind of weird partners in science there. All right, how do you map? We have to find differences between the genome before we can map something, right? What we're trying to do is say there's a trait that exists, right? Maybe it's a disease state or a phenotype of some sort, and you're trying to find where is the gene that codes for that on the genome, right? Now, if the genomes were all identical, we couldn't tell, right? And if the genomes only differed just because of that single trait, that would be virtually impossible to find. If you're looking for a single nucleotide polymorphism, right? The difference in having a trait and not having a trait is whether you have an A or a T at a certain position. That would be a single nucleotide change. If that was the only change that existed in the genome, you're looking for one base pair in 3 billion base pairs, right? There's no other way to find that single trait than to just sequence through every single nucleotide and try and find where the difference is. So what we have to do is rely on existing differences in the genome, things that we already know that exist in the genome, and see if your trait maps close to it. So the whole idea in physical mapping and genetic mapping in humans is trying to find a location in the genome that you already know differs between individuals, and see how close your trait is linked to that. Okay? So we're doing linkage analysis, but you can't generate the numbers and the statistics that you did in Drosophila. So you need to find polymorphisms. This is what I mean by places that the genome differs between individuals. Okay? So we're going to use existing polymorphisms, that's rare differences in the genome. Each individual in the human population is about 99.9% identical in terms of nucleotide sequence. Okay, so these are very rare events, less than a fraction or less than 0.1%. But there are these rare differences, and because of the Human Genome Project and because we've been doing genetics in humans for so long, we know where these polymorphisms exist in the genome. So what we're going to do is take these known places that vary, see how close your trait is to those known places. Okay? So we're going to take advantage of human recombination rates. Right? The way you're mapping in humans is basically the same way you're mapping in flies. You're just trying to see how often does a recombination event happen between the trait I'm looking for's gene, right? There's a gene for this trait, and it exists somewhere on the genome. And you want to know how many recombinations are happening between it and these known places on the genome. So it's mapping, right? There's a location of your gene on a chromosome, and then we're trying to find something that lies close to it because it's recombining, or actually because it's tightly linked and not very often recombining. We're looking for linkage between known polymorphisms and your trait. The human genome, we've been studying the human genome for a long time and trying to see what the actual recombination rate is. The recombination rate in the human genome is about 1.2 centimorgans per megabase. Okay, so remember, centimorgans is the percentage of times recombination happens between two locations on the genome. Okay, so this is a 1.2% chance of recombination every 1 million base pairs. Right, 1 megabase is 1 million bases, and the human genome is 3 billion base pairs long. So you actually have to do. There's a lot of work to be done. You have to look at a lot of meioses, a lot of chances of recombination happening to get linkage. Okay. So what we're looking for is a known polymorphism, and if a known polymorphism, a known sequence that differs between individuals, is always going with your trait, we would know that they're linked together, right? Every time you see an individual with this trait and you sequence their DNA, and they have that nucleotide sequence, then we'd say that there's a high level of those two being linked. If we know where the linked marker is, where this polymorphism exists in the genome, we know that that location on the genome is close to where the gene is that's coding for your trait, right? Yeah?
With the polymorphisms, are you saying that is the sequence that, let me ask the question this way. In the genetic difference or similarities between like us and mice or a mouse,
yeah,
there's some similarities in our genome.
Yeah.
Is the polymorphism the difference that between us and the mice?
Well, I'm saying the difference between you and I in terms of our genome, right? You and I differ at locations in our genome where you'll have one sequence and I'll have a different sequence. We know where there are common places in the human genome that people differ. Right? Over the vast majority of our chromosomes, we're 99.9% identical. But those fractions, those rare cases where we actually do differ, since we've been doing human genetics for so long, we know where those places are. We know that there is a place on chromosome 1 that has a high tendency to vary in individuals. And we know that there's a place on chromosome 2. So we have these genetic markers, they're landmarks that we know they exist on the chromosomes. And we're going to see how closely is that landmark linked with the trait that I'm thinking about. Right? If that sequence on chromosome 1 is always going with an individual that has your trait, you'd say, well, the gene that causes that trait is probably located close on chromosome 1 to that genetic marker. Okay? So we're talking about differences between individuals in a population. Yeah. Yeah.
So is this like it's kind of by chance then, because arbitrarily there can be multiple places in multiple chromosomes that
Yeah.
can change due to change in response to a trait that is displayed, not in response, but do you see what I'm saying? Like,
No, not quite.
There can be a trait between you and me that
Yeah.
I portray, but there can be differences between, there can be different polymorphisms within each chromosome
Yeah.
that we know are distinct.
Right.
So how do we?
Yeah, well, that's what the purpose of this lecture is. Yeah. So I will tell you what these polymorphisms look like and how you link them together. Yeah. But you're right. I mean, you have a couple of different alleles, and I have a couple of different alleles, and how do we figure out which ones are linked? That's what's going on here. So before we do that, I just want to say one last thing about recombination rates. That recombination rate that I gave you, 1.2% per megabase, is an estimate over the entire human genome. But each individual chromosome has a slightly different chance of recombining, and locations on that chromosome have higher chances of recombining, right? So this is a human genome. This is the recombination rates. So here is chromosome number. So chromosome number 1 is the assembly size in megabases. It's about 245 million base pairs, chromosome number 1, okay? Don't worry about assembly size, physical size. That's differences based on how you sequence the genome. Centimorgan length, the genetic length. So this is now trying to tie how often a recombination event happens and the size of the chromosome, okay? So they've looked at a bunch of different genes on chromosome 1, and the genetic length is 270 centimorgans, right? So centimorgans and megabases don't line up exactly, right? There's not recombination rate for every megabase, right? So what happens is the recombination rate is the centimorgan length, the genetic length, divided by the actual physical size, okay? So there's slightly more than one recombination rate, recombination event for every million base pairs. Okay, so that's on chromosome 1, the recombination rate is 1.1. On chromosome 2, it's 1.0 or 1.07. The big chromosomes are going to tend to show the overall recombination frequency. Right? These are all pretty close to 1.2, right? They're big chromosomes. That's just naturally, inherently how often the human genome recombines, okay? But notice when you get down here, the numbers start going up. So I don't know what this is. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. Somewhere around chromosome 14 or 15, you start to see the recombination rate going up, right? Instead of 1.2, it's now 1.3. That's because these are smaller chromosomes, okay? Chromosome 1 through 10 are really big chromosomes, and so they recombined at a certain frequency. When you start getting to be a smaller chromosome, even though it's a smaller chromosome, it lines up in metaphase, and there are recombinases that sit down and force it to recombine. So it's kind of counterintuitive, but a smaller chromosome actually recombines more often per length, right? Because the two chromosomes come together, and we force recombinations when the two chromosomes come together. So if you have two big chromosomes coming together, they're only going to recombine a certain frequency. If you have two smaller ones, though, we're going to force those to recombine. And since they're smaller and they still have similar number of crossing over events, genes are going to look like they're close, or like they're further apart. Does that make sense? If you got two genes that are a certain distance apart on a small chromosome, they're a certain distance apart, but since it's a small chromosome and there's more recombination events that happen, they're going to look like they're further away. So the smaller chromosome you get, see we're getting all the way down to chromosome 22 here, recombination rate is 2.1 now. It's a smaller chromosome, but it recombines more frequently per base pair, right? Because we force them to line up, and even though they're small, we are forcing them to recombine, okay? So we force more opportunities to recombine than if they were longer. So now places on the chromosomes themselves will recombine more often as well. So like one arm of chromosome 1 might have a higher recombination rate than the other arm of chromosome number 1. The factors that determine this is going to be, so I've talked about the size of the chromosome, right? So the smaller a chromosome is, the higher the recombination rate is typically. But also, within the chromosome, the GC content makes a difference. If there's a high number of G's and C's, that typically means it's a lower recombination rate than an area that has A's and T's. Okay? So if one arm of the chromosome 1 has lots of G's and C's, it's not going to recombine as quickly. Things are going to look closer together. If there's a lot of A's and T's, then things are going to recombine more frequently and they're going to look further apart. Okay. Proximity to either the telomere or the centromere. If you're at the telomeres, you tend to have high recombination frequency because you're kind of far out on the chromosome, and there's lots of cases of recombination at the far ends. If you're really close to the telomeres, there's not a lot of recombination that goes on really close to the telomeres. So low recombination rate there. I'm sorry, centromeres. Telomeres is high, at the ends of the chromosomes high. When you get to the middle of the chromosome, in the centromere, there's fewer recombination events. Repetitive regions. Repetitive regions mean that there's more homology, right? If you've got lots and lots of repeats, then those repeats find each other more often when the two homologous chromosomes line up. And so repetitive regions tend to increase your recombination rate. There'll be more recombination events there. Mutation rate also plays a role in it. If there's a region that's highly polymorphic, it differs a lot between individuals, then you might not be able to identify those as homologous chromosomes, right? If the sequence is really different on the two chromosomes, then you probably won't have recombining events there because the apparatus that brings those together is looking for homologous chromosomes. So if the sequences are very different on the two chromosomes, you won't have recombination. There's actually tricks in Drosophila that they do, where Drosophila has a weird genome where things can move around and parts of the chromosome can actually flip and invert themselves. And they will take regions and get them inverted. If you invert the region, it's not going to look the same anymore to the other chromosome. So you can basically stop a chromosome from recombining by using one of these inverted chromosomes. That's advanced genetic techniques, but if there's no similarity, you can't recombine, right? So rarely is X going to recombine with Y because there's very limited similarity. If you've got an X that's very different in a region than the other X, then that part's not going to recombine. This leads you to sex as well. Obviously, there's very little recombination between X and Y, so the Y chromosome virtually, we didn't even put the Y chromosome on this list because what is a Y chromosome going to recombine with? If you're a male, you've got an X and a Y. Those are the ones that you line up during meiosis, and there's no recombination between them. Now, the X has a recombination rate because in females, they have two X's. Those can line up, and you can have recombination. But there's really no recombination on the Y. So virtually zero. We don't even include it in the analysis. OK, so that's recombination rate. I just want you to know it varies between chromosomes, and it will vary at locations on the same chromosome. So we're going to talk about three mapping techniques, and I need to, though, make a distinction between physical map and genetic map. A genetic map is just a recombination frequency. Like what you did in flies is you take two traits and you say, how often does recombination happen between them, right? And so recombination frequencies in genetic maps, or the distance between things in genetic maps, is recombination rates, centimorgans. A physical map is how far are two genes apart in number of base pairs on the chromosome. Say, this gene lies at this position on the chromosome, and it is a certain number of base pairs away from another position on the chromosome. Okay. So typically what we want to do is get to a physical map. You want to know where on the genome is this gene located, because we're not so interested in like how tightly are things linked together typically. We're interested in what is the gene that's causing this disease, right? So we need to find that position on the genome, look at the protein that's being made or the mRNA that's being made, and decide at the cellular level what's happening, right? So in human mapping, we're almost always trying to get to a physical map. The way to do that is you have to link to these polymorphic regions, these anchors, these locations on the genome. And I'm going to talk about three different typing methods, okay? So the first one I'm going to talk about is called microsatellite mapping. This is repetitive regions in the genome. The second I'm going to talk about AFLP. This is called amplified fragment length polymorphisms. This is looking at different size pieces when you cut the genome. Okay? So Microsatellite is repetitive region. How long are the repeats that you have? AFLP is looking for when I cut the genome, what size pieces do I get? And I'll talk through these as I go. We'll talk about each one of these individually. The third one is single nucleotide polymorphism mapping. That's where I'm looking at differences of just a single base pair that differs between individuals. Okay? And I'm going to talk about these in this order. This is usually the order that you have to do to try and find a gene on the human genome. You can't just go straight for looking for single nucleotide polymorphisms, right? Because like we said before, you'd have to just sequence every single nucleotide in the disease individual and a normal individual and compare every single 3 billion nucleotides, right? You can't do that initially. You have to start out with more generic types of mapping, like see kind of what region is it, locate it to a chromosome, locate it to an arm of a chromosome, locate it to a position on an arm in the chromosome, and then maybe you can start looking for your polymorphism at the nucleotide level, okay? So these two are kind of generic methods. They're just looking for sizes, and we're not really concerned too much with the actual sequence. Single nucleotide polymorphisms, we're actually going to have to look at a sequence, okay? And this is just a little illustration of I think this is actually a Drosophila chromosome. But here is a picture of a Drosophila chromosome. You can see the banding pattern. We've represented that chromosome as little bands on this illustration. And then this is where they've actually located, here's a certain gene. It actually is located on that region of the chromosome. So this could be generated by a genetic map. But then when we actually tie it to a physical location on the chromosome, that becomes a physical map. All right, microsatellites. Microsatellites are repetitive pieces of DNA. And they are sporadic, that we find them on every single chromosome, and they're just, like Mark said, they're kind of random places around the genome. They could be functional. There are certain repetitive regions that are very important and functional to keep a chromosome in its right structure and its right state. But for mapping purposes, we're just going to think of them as random spots around there. And what they do is, since they're repetitive, they can differ in how many repeats exist. Okay? So it's repetitive region, and each individual, people differ at the number of repeat elements that they have. They're called microsatellites because the repeat sequence is a really short micro sequence. Okay? The sequence could actually be 2 to 6 base pairs in terms of what is the thing that's repeating. It might just be 6 base pairs long. In this example, this is a 3 base pair repetitive region. So here is CTT, and then it repeats CTT, CTT, CTT. So the repeat sequence is only 3 nucleotides. But in this individual, we've sequenced them, and they contain 8 repeats, okay? These are also called simple sequence repeats, so SSRs, or short tandem repeats. Since they're short and then they just repeat right after one another, we call them, the repeats are in tandem. So these are called STRs. These are all the same thing. So if you see microsatellite, SSR, STR, we're talking about the same repetitive elements, okay? Now, this individual has 8 repeats, but this individual has 7 repeats here. So it's repeated 7 times. This is how individuals vary at these microsatellites. Everybody has repetitive region at this location in the genome, but how many number of repeats you have varies, right? You inherited a certain number of repeats from mom and certain repetitive number of repeats from your dad. So your 2 chromosomes, probably at every single location on your genome, you probably have 2 different numbers of repeats at these, and we know about 10,000 known places in the human genome where these repeats regions exist. So we've got a lot of markers then, 10,000 known places in the genome, and you can then sequence people and just look for how big. You don't even have to sequence it really. You can just PCR amplify. And actually that's my next slide. The way you do this is you design a left PCR primer and a right PCR primer in the non-repetitive region that flanks it, okay? You wouldn't want to actually make your PCR primers for the repeat because they could sit down anywhere in that repetitive region. So you make the primers that flank it on the outside. You do PCR, and you allow those primers to amplify this region. And this guy has 6 repeats. This chromosome has 10 repeats. So you can take an individual, you just isolate DNA from them. You just take a small blood sample, isolate DNA, and then you can do PCR for any one of these 10,000 places in the genome. We've got primers, and you can just go order them. You can order primers that people have already made for any one of these 10,000 places in the genome. You can just use those primers, take the DNA from your affected, your population that you want to look at, and you just, do the PCR. You run out the products on the gel, and this individual is homozygous for the 6 repeats, right? When you run out the PCR sample on your gel, you only have 1 band, and it's a smaller band. So it's got 6 repeats, and since there's no second band, you know that both alleles, both chromosomes, have 6 repeats on them. So this is a homozygous individual for the 6 repeat. This individual is homozygous for the 10 repeat allele, and this is a heterozygous individual, right? So if you're looking for a human trait in like what causes color blindness, I don't know, pick a trait, whatever your gene is that you're interested in. What you do is sequence every single individual for a bunch of these microsatellite regions and see if your trait ever becomes linked to one of these microsatellites. Right? If every time you do the sequencing and what we're talking about, color blindness, if every colorblind individual always comes up with this allele, the 6/6 allele, then you'd say the gene that's causing color blindness must sit very close, it's tightly linked to this location in the genome, right? And we know where these microsatellites exist. This could be a microsatellite that sits on position 10,800 on chromosome 1, right? And we'd know whatever gene's causing color blindness is close to that arm of the chromosome number 1. Does that make sense? Now you can't do a bunch of crosses, right? You just have to go out into your population and find a bunch of colorblind people and do this sequence analysis on them and see if one allele seems to segregate with it, seems to be linked to it. If this one doesn't, then usually what you do is you take an individual's DNA, you put it on a 96 well plate. So you put 96 wells all containing the DNA from a single individual, and you do 96 microsatellites locations all in one swoop. You run out all of those 96 genotypes, and you see, does any one of these 96 places on the genome correspond to the color blindness trait? And if it doesn't, then you set up another plate and you do another 96 microsatellites until you've screened through, all 10,000 or whatever locations on the genome that we know. If you're lucky, your trait is only being coded for by one gene, and you find a linkage area. Sometimes, this is what makes human genetics so difficult, as we talked about a couple of times ago. Most traits are not just single genes, right? You could have epistasis effects, or multiple alleles, or X inactivation, or all these other attenuating circumstances that might make it hard to find just one location in the genome. Yeah.
So once we find the microsatellite region that does correspond to a trait, do we use restriction enzymes to cut out that fragment and then run more cycles of microsatellite mapping? Or
well, I'll talk. Yeah, eventually you want to identify the gene. So microsatellite is just kind of trying to get you into some region on the chromosome, right? Then you're going to focus in more closely on that region of the chromosome. Okay. Actually, I'll segue into what kind of the next step is. But first I want to make sure that everybody's clear on how microsatellites work. This is actually an illustration of a chromosome. And here are the known microsatellite regions there, right? So here's 0, here's one end of the chromosome. And we know that there are places all along, right? So for every chromosome, we know of these microsatellite regions. So if your trait is linked to one of these, it'd say like, oh, it's linked to that guy. Well, it must be close to that location on the genome, right? So then I'm going to zoom in there, and I'm going to do more mapping and see if we can narrow it down to a smaller region. Because this might only get you within, several megabases. If you're within, 4 megabases, that's still 4 million base pairs that you're going to have to look through, right? So, yeah.
I was just trying to understand how this helps you know that what we're looking for, like you're saying, color is near it. How does that indicate that it's near it?
Because that allele always goes with the mutant trait. So we're saying whatever allele is going with the mutant trait, we'd say that allele is on the same chromosome and tightly linked because there's very few recombination events that happen between them. It's just saying recombination frequency, basically. But it's saying recombination frequency to a certain location on the genome, right? We've already got a marker, a landmark on the genome. We say that's a microsatellite region, it exists right there, right? And if I sequence 100 individuals, and every individual that's diseased has an allele similar, has a common allele to that trait, I'd say then that allele, that microsatellite, and the allele that's causing the gene are very close together because I hardly ever see recombination. Every individual that's showing the trait always has this one microsatellite allele. I'd say that's because they're closely linked together, right? Did you have a question?
Sort of explain it.
Oh, okay, awesome. Can you articulate your confusion?
I understand to the point that we take like the population DNA,
Mm-hmm.
people that let's say have colorblind, and then we're trying to find that one region where that's the mutant allele,
Mm-hmm.
and then compare it to yours, and if you do have it, it's likely that you have
Yeah. So we're looking for a location on the genome. So like this microsatellite region, for instance. We're looking to see in affected individuals and non-affected individuals, do they have certain alleles, certain versions of that repetitive region, right? That repetitive region right here could either be 10 repeats long or could be 6 repeats long, right? There's different alleles there. And if everybody that has the trait always has the 6 repeat allele, then you would think that is because the gene that's causing the trait is very close on the chromosome to that allele, that 6 repeat allele, and there's no recombination between them. If what you did is sequenced everybody, and everybody that has the trait, some people are homozygous for the 6 repeat, some people are homozygous for the 10, and some people are heterozygous. And this 1/3, 1/3, 1/3, or I guess 1 to 2 to 1, you would say, well, these are randomly assorting, right? The trait is not at all linked to this location because everybody that's affected could have any one of the alleles on the other trait, right? But if everybody that has that trait is only showing this one allele, then you'd say those two must be close together on the genome because there's no recombination. Those traits are linked together. So we're saying our unknown trait, and we just have to look at the phenotype, right? Is the person colorblind or not? Is that trait of being colorblind linked to one of the alleles at this site? If they are, then we say that it must be close to that place on the genome, right? I don't know how to explain it any clearer than that. Maybe homework problems will help. If you have evidence of linkage to a certain location, it's probably going to be a huge region, right? Because the microsatellites only exist in certain locations on the chromosome. So you may have determined that your gene is linked somewhere between these two microsatellite regions, right? You've got linkage on either side of those, so the gene must exist in the middle. But that might still be, millions of base pairs wide, okay? The way you can then narrow this down is by a technique called restriction mapping. This is going to be where we cut the genome up into pieces. And again, we're just going to be looking for linkage. Is your trait, color blindness or whatever, linked to a certain piece size that we generate when we cut the genome? Okay. The way you cut the genome is with enzymes called restriction endonucleases. A restriction endonuclease is oftentimes just abbreviated and called a restriction enzyme. It's an endonuclease, so it cuts nucleotides. That's the nuclease part. It's an endonuclease because it cuts right in the sequence, right? We talked about exonuclease activity, the 5' or 3' exonuclease activity of polymerases. That's when they chew away at the ends of the DNA, right? Exonuclease. An endonuclease is when it cuts right in the middle of a sequence, okay? So you don't have to have a new polymerizing strand or anything. You just go right in, cut double-stranded DNA in the middle. It's a restriction endonuclease because it's restricted to cutting at only certain nucleotide sequences. So it's going to recognize certain gene sequences, somewhere between 4 to I don't know, dozens of base pairs long, but it will recognize a certain sequence and cut only when it finds that sequence. Okay. Mark and then Will. Yeah.
So you're saying, I just want to confirm, like, we have, so 99.9% of our genome is contingent with other people.
Yeah.
And the 0.1% is usually repetitive, like it's either SSR or STRs that, or are those
no.
the only sections that we're looking at?
Those are the most convenient sections to look at. So other repetitive regions differ between us, but some of that repetitive region is really big and they're really rare in the genome. The microsatellites are convenient because they're kind of spread out all sporadically, and so they give us a nice handy landmarks around there. But yeah, your genome is going to differ at those small repeat regions, big repetitive regions, single nucleotide changes. I mean, there's insertions and deletions. There's all kinds of ways that your genome differs. The microsatellites are just one of the ways, and they're just handy because they're spread all over the chromosomes. So.
And we're just using those as markers, right?
We're just using those, yeah, they just happen to be convenient markers, right?
So
Yeah.
we find the specific place where we think that the trait might connect to using the microsatellite plan. But then because there's still so many in there, we're using restriction mapping to,
Right.
again, zoom in or focus.
To zoom further in, yeah.
Okay.
Because if you're doing microsatellites, you're limited to where the microsatellites exist in the genome, right? So if I link it to right in between here, I can't do any further linkage other than saying it's somewhere between these two microsatellites, right? So how do I know? There could be hundreds of genes in between those. How do I zoom in and actually find the individual gene? Okay. So in in restriction mapping, we're going to cut at certain places in the genome. Oh. So here is an example of of restriction mapping. This is the normal globin gene. This is beta globin. It's one of the proteins that make up hemoglobin. This is the normal allele, normal sequence of a functional hemoglobin gene. It's got sequences right here called the DDE1 sequence. That's just a really short nucleotide sequence that this enzyme, the enzyme DDE1, recognizes, okay? So what that is saying is there's a sequence here that the enzyme will come in and recognize, and it's going to cut the chromosome right there, okay? So a normal version of the globin gene has 3 cuts here. One cut is in the non-coding region. 2 cuts exist in the coding region, right? So the colored region is where you're actually making the amino acid sequence, right? Here is a mutant version. This is the sickle cell allele, and it's different in its sequence where that DDE1 sequence is, okay? So whatever the mutation is that's causing the sickle cell mutation has disrupted the sequence that enzyme recognizes, okay? So if you cut a normal allele, it's going to cut 3 times, and it's going to give you this 200 base pair piece and this 175 base pair piece. In the mutant, I'm missing that sequence, right? That sequence is different, so I've lost that position where it's going to be cut. So instead of having 2 pieces, I get one big 376 base pair piece. And so if I take 2 individuals and cut them this way, you can determine what their what their alleles are, basically, right? We're looking at a normal allele that does cut, a 2-cutting allele, and a single, or this would be a 2-cutting allele for a single band. This would be a 3-cutting allele for 2 bands. Right? So this individual here has got 2 of the big alleles. That person has sickle cell anemia. This is normal, but it's a heterozygote, right? Normal phenotype. They've got, a normal homozygote. Both of their alleles are the normal one, so they cut twice on both chromosomes, so you just have 2 bands. This is a carrier, a heterozygote. So one of their chromosomes is normal, giving the 2 bands. The other chromosome is mutant, giving the single band. Right? So I'm showing you this in an example of a gene we already know. But again, this can be used exactly the same as microsatellite. Right? All we're doing is seeing, does your trait link to some certain size piece? Right? So I can use this the same way, but now I could actually use my restriction enzymes that are known to cut in the region you're looking at. Right? You could focus in and just look at the region that is between these 2 microsatellites. You isolate that region of DNA, and then you cut it and see what different cut sites you can find. So there's a bunch of these. There's hundreds of restriction enzymes, and they all cut at slightly different sequences. So you can use a restriction fragment length to zoom in on a smaller location within that. The same technique. Isolate DNA from your individuals in your population, isolate that region of the chromosome that you think is linked, and then cut it up with a bunch of different restriction enzymes and see if anybody who's showing that trait is linked to a certain one of these alleles.
So does that mean that the change is now within that 376 base pair length then, if we're looking at sickle cell?
Mm-hmm. Mm-hmm. What we're looking at is, the thing that's actually caused this is probably a single base pair change that just makes that this enzyme can't recognize the sequence anymore because one of the nucleotides is different. But you don't even have to know what's going on, right? For restriction enzymes, you don't have to know that it's associated with a certain mutation. What I'm just showing you is this is a way to look at different alleles, right? So you can just take that portion of the genome, you just cut it up with restriction enzymes, and you generate a bunch of these random-sized bands. We just say, do any of these random-sized bands always go with my disease trait? Then I would say, well, whatever's cutting, that cut site is close to our disease gene. Okay? So you can use restriction mapping. Amplified fragment length polymorphism, what I said at the beginning, it's just a version of restriction mapping. Basically, it's just restriction mapping, but then you use PCR to amplify it. So here is my genomic piece. Here is sequences that are cut by restriction enzymes. So I would take my DNA sample, I would digest it with these restriction enzymes so it cuts, and I make these fragments. AFLP just means you design primers for PCR that amplifies and makes more copies of all these little pieces. Okay, so it's restriction mapping, but it's just restriction mapping plus PCR to make yourself a lot of product so you can see it. This is what restriction mapping looks like. This is actually the restriction mapping that I did to find the mutant gene in the C-squirt for my PhD analysis. So this is actually an RFLP gel. So I took DNA, I cut it up into little fragments, amplified all those fragments by PCR, and then ran them out on a big gel, really tall, long gel. And so you can see all these tiny little bands that's showing up. The resolution's not good, but there's a tiny little band there, and a band there, and a band there. This one's everybody's got the same band there. So you can see some, and this is 3 mutant individuals, and this is 3 normal wild type individuals. And so you can see some things, the wild types and the mutants all have that allele. Sometimes just a single individual has an allele. Here's a single individual with this allele. I can't do anything with that. But if you find an allele, and here's 1, 2, 3, all the wild types have this allele, and all of the mutants are missing that allele. That seems to say that the wild type phenotype is linked to this location. So now I've got a location on the genome. I can take that piece of DNA I can go back and sequence that and find out where that sits on the chromosome. And I know my mutation never shows up with that allele, so the wild type allele must be linked to that region, and so conversely, the mutant allele is linked to that region as well, right? You can do this, you can map either way. But never does the vagabond or the mutant phenotype ever show up with this allele. That means it must always be segregating with a different allele. So I know that's probably a linked region, okay? You can take that and sequence it and narrow down then to an even smaller region on the genome. Go to the genome. We'll be done in just a minute here. This is human chromosome 2, and this is a 10 megabase region, right? A restriction mapping might still only get to a, still a fairly large piece. Here is a 10 megabase region in the human genome. All these red pieces are known genes, 35 known genes, and there's about 24 non-coding RNAs present. So all these guys in pink are non-coding RNAs. So I've got 50 or 60 potential candidates for my diseased allele at this point. And I'll talk about next time how you'd actually start testing every single one of those to see which one is actually your mutant gene. Okay.
Biola University offers a variety of biblically centered degree programs, ranging from business to ministry to the arts and sciences. Visit biola.edu to find out how Biola could make a difference in your life.